Today, I'm going to walk through the mechanism of action of Bepirovirsen, an investigational therapy for chronic hepatitis B that's designed to move patients toward what many trials call a functional cure, meaning sustained HBV DNA suppression with loss of hepatitis B surface antigen, HBsAg, after a finite course of therapy. First, what is Bepirovirsen? Bepirovirsen is a synthetic antisense oligonucleotide, or ASO, designed to bind HBV RNA transcripts inside infected liver cells and related liver-resident cell populations. Conceptually, it's a gene expression silencer for HBV, aimed upstream at the viral RNAs that drive production of viral proteins, including HBsAg. Now, let's break the mechanism into two major layers: a direct antiviral RNA targeting effect and a likely immunomodulatory effect that may help clear infected hepatocytes. I'll cover both because the best current evidence suggests Bepirovirsen is not purely a turn-down RNA drug, it may also be an activate immune clearance drug. Direct antiviral layer. Step 1. Tissue uptake and intracellular access. After subcutaneous dosing, Bepirovirsen distributes to the liver, where it can enter cells and reach the compartment containing HBV RNAs. Step 2. Sequence-specific binding to HBV transcripts. Bepirovirsen is engineered to hybridize, base pair, against a targeted HBV RNA sequence. This includes RNAs that contribute to HBsAg production, which is central because HBsAg is not just a marker, it's also thought to contribute to immune exhaustion and immune tolerance in chronic HBV. Step 3. RNA degradation and reduced viral protein production. When the ASO binds its target RNA, the RNA becomes vulnerable to enzymatic degradation, thereby functionally reducing the pool of HBV transcripts available for translation. Downstream, this is expected to reduce production of viral proteins, including HBsAg, and to reduce circulating HBsAg levels over time. Clinically, the signature of this direct layer is declining HBsAg during treatment, and in some patients, HBsAg loss that can persist after therapy stops, part of what functional cure endpoints are aiming to capture. 2. Immunomodulatory/immune clearance layer. Now the second layer, immune activation and infected cell clearance. Several datasets suggest Bepirovirsen can trigger innate immune signaling, with patterns consistent with activation of inflammatory cytokines and immune pathways. One proposed mechanism is TLR8-mediated innate immune activation. Why does that matter? Because chronic HBV is not just a virology problem, it's an immunology problem. Long-standing high antigen loads, especially HBsAg, are associated with immune dysfunction. So a therapy that both reduces antigen and nudges innate immunity could, in theory, improve the probability of immune-mediated control. In biomarker analyses, Bepirovirsen treatment has been associated with immune activation signatures and with patterns suggesting hepatocyte injury and death occurring more often in virologic responders, raising the possibility that some ALT flares might reflect immune-mediated killing of HBV-infected hepatocytes rather than nonspecific hepatotoxicity. Importantly, this is still an evolving story. Immune activation appears to occur, but the exact causal chain, how much is required for response, who benefits, and what the safest therapeutic window is, remains an active area of study. This brings us to a clinical pattern that many clinicians notice: ALT elevations during therapy. In the Phase 3 program, Grade 3 or higher adverse events occurred more often with Bepirovirsen than placebo, and ALT increases were the most common Grade 3 adverse event reported with Bepirovirsen. Mechanistically, that fits with two non-mutually exclusive possibilities. ALT rises as an on-target signal linked to immune engagement and infected hepatocyte clearance, and/or ALT rises as an inflammatory effect that accompanies innate immune stimulation in the liver. Either way, the key mechanistic point is that ALT elevations may be intertwined with the drug's overall biology, so they're not automatically interpretable as classic dose-dependent drug-induced liver injury without context. Resistance and viral variation. A natural question with any sequence targeting therapy is, what about HBV sequence polymorphisms, Does mismatch lead to resistance? In a phase 2b analysis evaluating single nucleotide polymorphisms in the Bepirovirsen binding site, baseline SNPs were identified in a minority of participants, and most non-responders or relapsers did not harbor binding site SNPs, suggesting that sequence mismatch is not the dominant driver of non-response for most patients. In vitro, certain variants can reduce susceptibility, but overall, the available evidence suggests routine pre-treatment variant testing, specifically for the Bepirovirsen binding site, is not currently warranted, at least based on what's been reported so far. How does this fit into combination therapy and stop-to-cure strategy? Finally, it's important to place Bepirovirsen within modern HBV cure strategy design. In the phase 3 trials, patients were on stable nucleoside analog therapy, received fixed-duration Bepirovirsen, and then nucleoside analog therapy was discontinued in eligible patients later in follow-up, testing the idea that you can reduce antigen burden and potentially reshape immune control, then attempt durable suppression off therapy in responders. Mechanistically, that strategy relies on Bepirovirsen doing more than transiently lowering HBV DNA, because nucleoside analogues already do that. The value add is the potential for HBsAg loss and sustained off-treatment control in a subset. So, in summary, Bepirovirsen appears to work through a multimodal mechanism: direct, sequence-specific targeting of HBV RNA transcripts, reducing viral protein production, especially HBsAg; innate immune activation, with evidence consistent with TLR8-linked signaling and broader immune engagement; and potentially immune-mediated clearance of infected hepatocytes, where ALT elevations may sometimes function as a biomarker of biologic activity rather than purely off-target toxicity.